A footprint, a broken skeleton, a stone struck against another stone. Each of these discoveries seemed small at first, yet every one forced scientists to rethink where we came from, how we changed, and whether our species was ever truly alone. These are 10 fossil and archaeological discoveries that rewrote human history. Number 10. The Laetoli Footprints. In northern Tanzania, not far from Olduvai Gorge, a trail of footprints crosses a layer of hardened volcanic ash. There are no bones beside them, no tools, no remains of a camp, only a sequence of impressions left by several individuals who walked across a damp landscape roughly 3.6 million years ago. Soon after they passed, more volcanic material fell over the surface. The tracks were sealed beneath new layers and preserved for millions of years. When researchers eventually exposed them, they found something far more important than a record of where a few ancient creatures had walked. The prints preserved how they walked. The main trail extends for nearly 88 feet and contains about 70 early human footprints. Their makers appear to have placed the heels two down first, shifted their weight forward, and pushed off through the front of the foot. It was a stable, repeated form of walking on two legs. This was not an ape briefly standing upright. It was habitual bipedal movement. That distinction changed the timeline of human evolution. The individuals who crossed the Laetoli landscape lived long before the appearance of the genus Homo. Their brains were still much closer in size to those of living apes than to ours. They had no cities, no writing, no metal. Yet one of the defining features of the human body was already present. Fossils of Australopithecus afarensis have been found in the same region and geological period, making that species the leading candidate for the track makers. But footprints do not preserve faces. They contain no DNA. The exact identity of the walkers cannot be proven with complete certainty. What can be established is more important. By 3.6 million years ago, at least one early human population was already moving upright across the ground, with a surprisingly human-like stride. For a long time, it was easy to imagine that human evolution began with intelligence. A larger brain supposedly led to better tools, more complex behavior, and eventually upright walking. Laetoli showed that the sequence was different. Walking came first. The large human brain arrived millions of years later. Why bipedalism evolved is still debated. Walking upright may have reduced the energy needed to travel between scattered food sources. It may have freed the hands to carry food or infants. It may have helped early humans reach higher branches or see across tall vegetation. There may never have been one single cause. Evolution often works through several small advantages acting together over long periods. Laetoli does not explain why our ancestors stood up. It gives us something more direct. It records the moment when they did. A fossil bone can suggest how an animal might have moved. A footprint captures an action that actually happened. Several individuals crossed wet volcanic ash, step after step, unaware that their movements would outlast their entire species. But those tracks reveal only what happened at ground level. They cannot show the shoulders, the arms, the spine, or the full shape of the pelvis. To understand the strange body behind footsteps, scientists needed more than a trail. They needed a skeleton. Number 9. Lucy. In November of 1974, paleoanthropologist Donald Johansen was surveying the dry landscape near Hadar, Ethiopia, when he noticed a small piece of bone on the ground. It was part of an elbow. Then more fragments appeared. A rib, pieces of pelvis, sections of the spine, bones from the arms and legs. By the end of the excavation, the team had recovered roughly 40% of a single skeleton. The fossil was assigned the catalog number AL-2881. The world would know her as Lucy. She belonged to Australopithecus afarensis and lived about 3.18 million years ago. Lucy was not the oldest human ancestor ever found. She was not even the first evidence of upright walking. Her importance came from how much of one body had survived. Before Lucy, researchers often had to reconstruct extinct human relatives from isolated jaws, teeth, or fragments of skull. Here, many parts of the same individual could be studied together. The pelvis was short and broad compared with that of an ape. The thigh bone angled inward toward the knee, positioning the feet beneath the body. The lower spine supported an upright posture. Lucy clearly walked on two legs. But the rest of her anatomy did not resemble a smaller version of a modern human. Her arms were relatively long. Her shoulders retained features useful for climbing. Other members of her species had curved fingers, suggesting that movement through trees remained important. This combination challenged a simple idea of human evolution. Lucy was not halfway between an ape and a person. She was not an unfinished version of us. She belonged to a successful species with its own distinctive way of living. On the ground, she could walk upright. In the trees, her upper body still offered strength and mobility. Those abilities may have complemented each other. Woodlands offered fruit, shelter, sleeping places, and protection from some predators. Open ground provided access to resources spread across larger areas. Australopithecus afarensis could move between both environments. Lucy's skeleton also revealed something deeper. Evolution does not transform an entire body at once. A new way of walking can appear, while the arms remain suited to climbing. The pelvis can change while the brain stays small. Teeth, hands, feet, shoulders, and skulls can follow different evolutionary paths. Scientists describe this as mosaic evolution. The human body was assembled gradually, through features that appeared at different times and for different reasons. Nothing changed according to a single plan. That is why words such as primitive and advanced can be misleading. A feature is not advanced simply because modern humans possess it. It is useful only in relation to a particular environment. Lucy's climbing anatomy was not a failed step toward becoming human. It helped her species survive. Important questions remain. Researchers still debate how much time Australopithecus afarensis spent in trees and how efficiently the species walked over long distances. The bones can narrow the possibilities. They cannot reconstruct every hour of Lucy's life. Still, the central lesson is clear. Long before the human brain became large, a small-bodied hominin was already walking upright across East Africa while maintaining a close connection to the trees. Laetoli recorded the steps. Lucy revealed the body behind them. Together, they changed how scientists understood human movement. But movement was only one part of the story. At some point, our ancestors began doing something different. They stopped simply using the world as they found it. They began deliberately reshaping it. For decades, scientists believed that this technological turning point belonged to the genus Homo. Then a discovery near Lake Turkana pushed stone toolmaking much deeper into the past. Number 8. Lomekwi 3. In 2011, a research team working west of Lake Turkana in Kenya took a wrong turn. The mistake led them into an area scattered with unusual stone fragments. Some appeared to have been struck deliberately. Excavations followed. Below the surface, researchers found heavy stone cores, sharp flakes, anvils, and battered objects. The site became known as Lomekwi 3. Its age was the real surprise. The artifacts were about 3.3 million years old. That made them roughly 700,000 years older than the earliest classic Oldowan tools known at the time. It also placed them before the oldest widely accepted fossils of the genus Homo then known to science. The traditional story linked stone tools with a growing brain. Members of Homo were thought to have developed the planning, coordination, and hand control required to strike one stone against another and produce a useful edge. Lomekwi complicated that idea. The tools were large and heavy. Some cores weighed more than 30 pounds. They were also different from the smaller, more controlled flakes associated with later Oldowan technology. The makers appear to have used several methods. Some stones were struck directly against other stones. Others were placed on hard surfaces and hit from above. Stationary rocks may have been used as anvils. The results were not random. Repeated impacts created recognizable patterns of fracture. Whoever worked at Lomekwi understood that stone could be broken in predictable ways. That does not mean the toolmakers thought like modern engineers. It means they recognized a physical relationship between material, angle, and force. A rounded stone could become a sharp edge. A heavy block could become a hammer. An anvil could become part of a repeated process. The identity of the makers remains unknown. No fossilized hand was found holding one of the tools. Kenyanthropus platyops lived in the broader region at around the same time. Australopithecus afarensis existed elsewhere in East Africa. Either might be considered a candidate. Another population may have been responsible. The evidence cannot choose between them. That uncertainty is essential. Lomekwi should not be used to declare that one species invented technology. Its significance lies elsewhere. Intentional stoneworking may have begun before the genus Homo and before the major expansion of the human brain. That possibility changes what archaeologists search for. If small-brained hominins could modify stone, then technology did not suddenly appear as a complete package. It may have developed from older behaviors already present in primates. Chimpanzees use stones to crack nuts. Other primates strike objects, carry tools, and learn local traditions. But breaking stone to create a new edge adds another step. The object is not simply being used. It is being transformed. The exact purpose of the Lomekwi artifacts remains unclear. Sharp flakes could have cut plants, meat, or hides. Heavy stones might have opened nuts, roots, or bones. Without clear residues or directly associated butchered remains, those uses cannot be confirmed. Even the interpretation of the site continues to be tested. A date this early demands careful attention to the position of the artifacts and the possibility that natural forces moved them. The excavators reported tools embedded within dated sediment, supporting an age of 3.3 million years. But scientific discoveries grow stronger through scrutiny. Lomekwi matters because it created a question that cannot be dismissed. If the artifacts represent deliberate technology, then the foundations of toolmaking appeared far earlier than expected. The first toolmakers may not have looked like members of our genus. They may not have had our hands, our brains, or our style of movement. Technology may have begun as something rough, physical, and experimental. A strike, a fracture, a result remembered and repeated. That is very different from a sudden moment of invention. But making tools in one location is not the same as surviving across an entire continent. For that, early humans required a body capable of moving efficiently through large and changing landscapes. Near another part of Lake Turkana, a nearly complete skeleton showed when that transformation had taken place. Number 7. Turkana Boy. In 1984, fossil hunter Kamoya Kimeu noticed a small piece of skull near Nariokodome, on the western side of Lake Turkana in Kenya. Excavations uncovered much more. Ribs, vertebrae, a pelvis, arms and legs. Most of the skeleton of a young individual slowly emerged from the sediment. The fossil became known as Turkana Boy or Nariokotom Boy. Its scientific catalog number is KNMWT 15,000. He lived about 1.6 million years ago and is usually assigned to Homo erectus, although some researchers use the name Homo ergaster for the African form. The classification matters to specialists. The body matters to the larger story. Earlier hominins such as Lucy were short, with relatively long arms, and many features connected with climbing. Turkana Boy was different. His legs were long. His arms were proportionally shorter. His body was narrow and tall. From the neck down, his general proportions were much closer to ours. That does not mean he was anatomically modern. He was still growing when he died. and his skeleton retained its own distinctive features. Scientists have also debated his exact age because early Homo may have matured faster than modern humans. He was probably in late childhood or early adolescence. Whatever his age, the skeleton provides one of the clearest views of the Homo erectus body. Long legs increase the distance covered with each step. A narrow trunk reduces the amount of body surface exposed to overhead sunlight. Long limbs can release heat efficiently. These proportions are useful in hot, open environments where water and food may be widely separated. Homo erectus was built for life on the ground. The species did not simply walk upright. It could travel. That ability may have changed the scale of human survival. A population tied to one small patch of woodland must depend on local resources. A population capable of moving long distances can follow water, animals, and seasonal plants. Mobility turns geography into opportunity. Turcanaboy's brain was larger than that of Australopithecus, but still smaller than the average modern human brain. Once again, the sequence was not what older theories might have predicted. The near-modern body appeared before the modern brain. Researchers have proposed that Homo erectus may have been capable of endurance running. Some features of the skeleton are consistent with improved long-distance locomotion. But exactly how often these humans ran, or whether they regularly chased prey over great distances, cannot be proven from one skeleton. The safer conclusion is already powerful. Their bodies were adapted to sustained movement on the ground. They could cover territory in ways earlier homins probably could not. The fossil also opened a window into growth. Turcanaboy was tall for his age. Had he reached adulthood, he may have stood well over 5 feet tall, perhaps closer to 6 feet depending on the growth model. Yet evidence from the teeth suggests that he matured faster than people do today. The extended childhood familiar to modern humans had not fully developed. His body was becoming modern in shape, while his life history remained different. This was mosaic evolution on another scale. Legs, brain size, childhood, social behavior, and technology did not change together. Each followed its own timeline. Homo erectus would become one of the most successful human species ever known. Populations connected with this broad group spread across Africa and large parts of Eurasia. But Turcanaboy raised a larger question. When did these travelers first leave Africa? And how advanced did they need to be before they could survive beyond it? At Dmanisi, in the Republic of Georgia, 5 skulls gave an answer no one expected. Number 6. Dmanisi. On a hill in the southern Caucasus stand the ruins of a medieval settlement. Beneath them are layers almost 2 million years older. Those deposits contain extinct animals, simple stone tools, and the bones of early humans who lived near the gateway between Africa, Europe, and Asia. The Dmanisi hominins date to roughly 1.8 million years ago. They represent some of the earliest direct fossil evidence of humans outside Africa. Before their discovery, researchers often pictured the first major migration as the result of advancement. Perhaps humans needed larger brains. Perhaps they needed sophisticated hand axes. Perhaps they required a body and behavior close to those of later Homo erectus. Dmanisi showed something different. The individuals had relatively small brains. Some cranial capacities overlapped with much earlier members of Homo. Their tools were simple. There is no evidence at the site of an elaborate technological system. Yet these populations had reached the Caucasus. The journey did not require a modern mind. It did not require modern equipment. It may not even have felt like a journey. Populations could expand gradually over hundreds of generations, following familiar animals, plants, and environments. No individual needed to know that a continent had been left behind. A movement of only a few miles in each generation could eventually carry descendants enormous distances from the land of their ancestors. Dmanisi is also important because it preserves several individuals from the same place and general period. 5 skulls revealed striking variation. One had a broad face and a small brain case. Another was more lightly built. The jaws and teeth differed considerably. Had those fossils been found at separate sites, they might have been assigned to different species. Because they came from one population, researchers had to consider how much variation could exist within early Homo. One interpretation argued that several named African species might represent regional or individual variation within a single changing lineage. Other researchers disagreed. They maintained that the early Homo fossil record still supports multiple species. The debate has not been settled. Denisova did not simplify early human evolution. It revealed how difficult the problem really is. Living populations vary because of sex, age, health, ancestry, and individual development. Fossil species are reconstructed from tiny and uneven samples. When a species is named from one jaw or a partial skull, its natural range of variation may be unknown. Denisova gave researchers something rare: several faces from one ancient community. One individual was especially revealing. An older adult had lost almost all of the teeth in the lower jaw years before death. The empty sockets had healed. Surviving without teeth would have made eating difficult. The individual may have consumed soft foods, used tools to process meals, or received assistance from others. The fossil does not prove a formal system of caregiving, but it shows that a severely impaired member of the group survived for a significant period. Life at Denisova was not simply a contest between isolated individuals. These early humans belonged to a social population. The site changed two parts of the story. First, humans left Africa earlier and with less advanced anatomy and technology than many scientists had expected. Second, early Homo may have contained far more variation than neat textbook diagrams suggest. Migration did not wait for perfection. It was carried out by adaptable, socially connected beings with modest brains and simple tools. Once outside Africa, populations could become separated by distance, climate, and time. Some disappeared. Others contributed to later lineages. But how early did the major branches of Eurasian humanity begin to form? Deep inside a Spanish cave, a concentration of bones preserved a population close to the roots of the Neanderthals. Then ancient DNA made the story even more complicated. Number 5. Cima de los Huesos. The name means pit of bones. It lies within the Atapuerca cave system in northern Spain. Reaching it requires traveling through narrow underground passages to a deep chamber where thousands of human bone fragments accumulated. The remains represent at least 28 individuals. They lived roughly 430,000 years ago. This is not one skull or one jaw. It is one of the largest collections of Middle Pleistocene human fossils ever found. The bones preserve a mixture of features. Some parts of the skull and teeth resemble later Neanderthals. Other characteristics appear more generalized. For years, researchers debated how the population should be classified. Some placed the fossils within Homo heidelbergensis. Others emphasized their early Neanderthal traits. The anatomy suggested that Cima de los Huesos stood close to the Neanderthal lineage. DNA seemed to challenge that conclusion. Recovering genetic material from fossils this old is extremely difficult. After death, DNA breaks into smaller fragments. Heat, moisture, microbes, and chemical reactions destroy the original molecules. Most ancient bones contain far more DNA from soil organisms and modern contamination than from the individual itself. The cool, stable environment of the cave created an unusual opportunity. Researchers first recovered mitochondrial DNA from a thigh bone. Mitochondria are structures inside cells that carry their own small genome. Because each cell contains many copies, mitochondrial DNA is often easier to recover than nuclear DNA. The result was unexpected. The sequence appeared more closely related to Denisovans than to Neanderthals. That did not fit the anatomy. The bones looked early Neanderthal. The mitochondrial DNA pointed toward a population known mainly from Siberia. For a time, the finding threatened to rearrange the human family tree. Then researchers recovered fragments of nuclear DNA from additional specimens. Nuclear DNA contains the much larger genome inherited from both parents. That evidence placed the Sima population closer to Neanderthals than to Denisovans. The anatomy had not been misleading, but the mitochondrial result still required an explanation. Several possibilities remain. The population may have retained mitochondrial ancestry from an older group that later disappeared from Neanderthals. Gene flow from another population may have introduced a different maternal lineage. Later demographic changes may have replaced one form of mitochondrial DNA with another. The exact history is unresolved. What became clear was that a simple branching diagram could not explain everything. A population can be mostly related to one lineage while carrying genetic material connected to another. Different parts of the genome can preserve different histories. The Sima evidence also pushed the Neanderthal-Denisovan split deeper into time. If individuals living 430,000 years ago were already closer to Neanderthals, then the separation between the Neanderthal and Denisovan lineages must have happened earlier. Eurasia was already divided among major human populations hundreds of thousands of years before Neanderthals disappeared. Cima de los Huesos presents another mystery. Why did so many bodies accumulate in one difficult chamber? Natural processes may have moved remains into the pit. Bodies may have fallen. Water, carnivores, or geological activity may have contributed. Some researchers have suggested that the dead were deliberately placed there. The evidence has not produced universal agreement. The importance of the site does not depend on solving that question. The bones transformed an abstract lineage into a population. Children, adolescents, and adults once lived in Western Europe. Their anatomy already carried the beginnings of the Neanderthal form. Their DNA preserved evidence of a deeper and more tangled ancestry. The family tree was beginning to look less like a tree. But the most surprising combinations were not limited to genes. In South Africa, another cave contained a human species whose anatomy seemed to combine traits separated by millions of years. More surprising still, it lived far later than anyone first expected. Number 4. Homo naledi. The Rising Star Cave system in South Africa is not an easy place to work. Some passages are so narrow that researchers must turn sideways and squeeze between the rock. Beyond one of those constrictions lies the naledi chamber. In 2013, explorers entered the chamber and found bones across its floor. A specialized excavation team was recruited to work in the confined space. During the first major phase of recovery, more than 1,500 fossil specimens were collected. They represented at least 15 individuals. The fossils were assigned to a newly named species, Homo naledi. Its anatomy was difficult to place. The skull was small. The brain was only a fraction of the average size of a modern human brain. The shoulders and pelvis retained features resembling much earlier hominins. The fingers were curved. Yet the wrist, hand, foot, and lower limb also displayed traits associated with more recent members of Homo. The body looked like a mosaic assembled from different periods of human evolution. At first, many researchers assumed the fossils must be extremely ancient. A small brain and primitive-looking anatomy seemed to belong near the beginning of the genus Homo. Dating produced a surprise. The remains were only about 335,000 to 236,000 years old. Homo naledi lived during the same broad period in which early Homo sapiens was emerging in Africa. That does not prove the two species met. No direct evidence demonstrates contact, but their known time ranges overlapped. Africa was not occupied by a simple sequence in which one form disappeared before another arrived. Different kinds of humans could exist during the same period. Some had large brains. Others did not. Some had bodies closer to ours. Homo naledi retained a distinctive combination of old and new. The discovery challenged a powerful assumption. Small-brained humans did not belong only to the distant past. Brain size is not a clock. A lineage can retain smaller dimensions while surviving beside lineages that have changed in other directions. The location of the fossils created another problem. The chamber is deep, dark, and difficult to reach today. How did the bodies get there? The relative lack of other large animal remains made a simple predator den less convincing. Researchers considered water movement, accidental falls, repeated entry, and deliberate placement. Claims have since been made that Homo naledi intentionally buried bodies, used fire inside the cave, and created markings on the walls. Those claims remain heavily debated. Other specialists argue that the evidence does not yet demonstrate burial or symbolic behavior. The distinction matters. A compelling interpretation is not the same as a proven conclusion. The secure discovery is already remarkable. A small-brained human species survived into the Middle Pleistocene and left an unusually large collection of remains deep within a cave system. Its hands could grip and manipulate objects. Its feet were well adapted to upright walking. Its upper body retained climbing-related features. Homo naledi did not fit a ladder from primitive to advanced. It occupied its own branch. That is why the species remains important, even if the behavioral claims are eventually rejected. Human evolution did not steadily produce larger brains and more modern bodies in every surviving population. Different lineages followed different paths. Some combinations endured much longer than expected. Rising Star revealed diversity within Africa. But even that did not prepare scientists for what emerged from an Indonesian cave. There, a small-bodied human with a tiny brain survived until tens of thousands of years ago, on an island that has never been connected to mainland Asia by dry land. Number 3. Homo floresiensis. Liang Bua is a limestone cave on the Indonesian island of Flores. Archaeologists were excavating there while investigating the movement of early humans through Southeast Asia. In 2003, they uncovered the partial skeleton of an adult. But the individual stood only about 3.5 feet tall. The skull contained a brain of roughly 400 cubic centimeters, comparable in volume to that of a much earlier Australopithecine. The specimen was labeled LB1. More bones and teeth from other individuals followed. This was not simply one unusually small person. Researchers announced a new species, Homo floresiensis. The public quickly gave it a nickname, the Hobbit. The nickname made the discovery famous, but it also risked making the species seem like a curiosity. In reality, Homo floresiensis presented one of the most serious challenges to simple models of human evolution. The original dating suggested that some remains might be as young as 12,000 years. That would have meant the species survived until long after agriculture began elsewhere in the world. Later research revised the geological interpretation. The sediments containing the fossils were older than first believed. The skeletal remains are now generally dated to between about 100,000 and 60,000 years ago. Stone tools at Liang Bua extend from roughly 190,000 to 50,000 years ago. The revised dates made the species older. They did not make it less important. Homo floresiensis still lived remarkably late for a human with such a small body and brain. The first major debate concerned whether LB1 represented a genuine species. Some researchers proposed that she was a modern human with a medical condition that affected growth or brain size. Several diagnoses were suggested, but additional fossils weakened that explanation. Multiple individuals shared small dimensions. Features of the wrist, shoulder, jaw, foot, and skull did not fit neatly within the range of a single diseased modern human. Most researchers came to accept Homo floresiensis as a distinct lineage. The next question was harder. Where did it come from? One hypothesis begins with Homo erectus. Homo erectus reached Java more than 1 million years ago. A population may have arrived on Flores and then undergone island dwarfism. On islands with limited food and few large predators, large-bodied animals sometimes evolve smaller forms. Flores was also home to dwarf relatives of elephants known as stegodonts. Perhaps humans responded to similar ecological pressures. Yet Homo floresiensis retained features that some researchers considered more primitive than expected from a dwarf, Homo erectus. The wrist, shoulder, foot, and parts of the skull have been compared with much earlier hominins. This produced another possibility. The ancestors of Homo floresiensis may have left Africa before the classic form of Homo erectus evolved, carrying older anatomy into Asia. The fossil record between Africa and Flores remains too incomplete to settle the question. Discoveries elsewhere on Flores have shown that small body size appeared early in the island lineage. Tiny teeth and limb bones from Mata Menj date to around 700,000 years ago. Some traits resemble early Asian Homo erectus. That supports an evolutionary connection, but does not explain every feature. However the lineage began, it had been on Flores for an immense length of time. Then there is the ocean. Even during periods of low sea level, Flores remained separated from neighboring islands by deep channels. The ancestors of Homo floresiensis had to cross water. That does not prove they built boats. Individuals may have been swept away on natural rafts after storms or floods. A deliberate crossing is also possible, though direct evidence has not survived. The journey remains one of the greatest mysteries surrounding the species. Stone tools show that these small humans manipulated their environment. Animal remains at Liangbua include stegodonts, giant rats, reptiles, and birds. Determining exactly which species processed which animals is difficult because the cave was occupied at different times. Still, Homo floresiensis was not helpless. A small brain did not prevent the lineage from surviving on an isolated tropical island for hundreds of thousands of years. Its disappearance is unresolved. The youngest known tools associated with the species date to around 50,000 years ago. Modern humans were moving through Southeast Asia during the broader period. Climate change, volcanic activity, ecological disruption, competition, or disease may have contributed. No evidence proves that Homo sapiens directly caused the extinction. The overlap may have been brief or indirect. The absence of younger fossils cannot tell us why a species vanished. Homo floresiensis changed the map of human evolution. A remote island once held a distinct human lineage with a body unlike ours, a brain far smaller than ours, and a history extending deep into the late Pleistocene. Asia had not been populated by one uniform wave of Homo erectus followed by modern humans. It contained isolated survivors, local experiments, and branches unlike anything alive today. While Homo floresiensis lived on Flores, our own species was developing in Africa. For much of the 20th century, scientists searched for a specific birthplace and a clear moment when modern humans appeared. A group of fossils in Morocco revealed that the origin of Homo sapiens was older, wider, and less sudden than expected. Number 2. Jebel Irhoud. Jebel Irhoud lies in Morocco, in northwestern Africa. Human fossils were first uncovered there during mining operations in the 20th century. The remains included skulls, jaws, and teeth associated with Middle Stone Age tools. At first, the fossils were believed to be much younger than they really were. Their anatomy was confusing. The faces looked surprisingly modern. The brain cases did not. Later excavations recovered additional fossils and clearer information about the archaeological layers. Researchers also used thermoluminescence dating on flint artifacts heated in ancient fires. The estimated age was about 315,000 years, with a substantial range of uncertainty. That placed Jebel Irhoud near the earliest known stages of Homo sapiens. The discovery did not reveal people identical to us. Their faces were relatively flat and reduced compared with many earlier humans. From the front, A person with a Jebel Irhoud face might not have appeared completely unfamiliar, but the skull was longer and lower than the rounded brain case typical of people today. Modern facial anatomy had emerged before the fully modern shape of the brain case. Once again, evolution had assembled the human form in pieces. This mattered because older models often placed the appearance of Homo sapiens in East Africa around 200,000 years ago. From one small source population, the species was then imagined spreading across the continent and eventually the world. Jebel Irhoud pushed important features of our lineage back by roughly another 100,000 years. It also shifted attention to North Africa. The origin of Homo sapiens could no longer be described as one event confined to one eastern or southern location. The researchers behind the study proposed a broader process across Africa. During wetter periods, populations in different regions could become connected by grasslands, rivers, and lakes. During dry periods, deserts and ecological barriers could separate them. Gene flow increased and decreased over time. Different populations may have developed different elements of modern anatomy and behavior. When connections returned, those traits could spread. Our species may have emerged from a network rather than one isolated birthplace. Other fossils add to this picture. Homo was discovered in southwestern Ethiopia and has long been considered one of the earliest clearly modern human skeletons. Dating the site was difficult because the fossils lay among volcanic layers that had to be matched across the East African rift. Later geochemical work linked the ash above Homo I to a major eruption of the Shala volcano. That eruption occurred roughly 233,000 years ago, with a significant margin of uncertainty. Because the fossil lies beneath the ash, Homo I must be older than the eruption. Jebel Irhoud and Homo I do not form a simple sequence from primitive to modern. They come from different regions and preserve different combinations of anatomy. That is the point. Early Homo sapiens was geographically and physically diverse. The species did not appear everywhere in one finished form. The face, brain case, body, behavior, and genetic structure continued to change. Archaeology tells a similar story. Middle Stone Age technologies appeared across Africa in varied forms. Pigments, long-distance movement of materials, specialized tools, and symbolic objects emerged at different places and times. There was no single morning when humans suddenly became behaviorally modern. Capabilities accumulated. Some disappeared. Others reappeared. Populations exchanged knowledge when contact was possible and developed local traditions when isolated. This interpretation changes the meaning of the word modern. A fossil can belong to the Homo sapiens lineage without possessing every feature common today. Species are populations evolving through time. They are not fixed designs. The people of Jebel Irhoud were not unfinished versions of us. They were humans adapted to their own world, carrying some traits that later became widespread and others that continued to change. The discovery answers part of the central question. Homo sapiens has deep roots across Africa, but it creates another problem. When members of our species expanded into Eurasia, they entered landscapes already occupied by other humans. Neanderthals lived in Europe and Western Asia. Denisovans occupied parts of Asia. Other populations may still be unknown. Did Homo sapiens simply replace them? For years, that seemed possible. Fossils showed different forms appearing and disappearing, but bones alone could not reveal every encounter. Then scientists extracted DNA from a tiny fragment in a Siberian cave. The result uncovered an entire population. A second fragment revealed something even more important. Those populations had not remained separate. Number 1. Denisova Cave. Denisova Cave lies in the Altai Mountains of southern Siberia. For thousands of years, the cave offered shelter to animals and different groups of humans. Its sediments preserved tools, ornaments, bones, charcoal, and traces of occupation extending deep into the Pleistocene. Among the recovered material was a small fragment from the end of a child's finger. Its shape revealed almost nothing. It could not provide a face. It could not show height, body proportions, or brain size. Researchers extracted mitochondrial DNA. The sequence did not match modern humans. That was not surprising. It also did not match known Neanderthal mitochondrial DNA. That was unexpected. A previously unknown human population had been discovered, not through the shape of a skull, but through genetic code. The group became known as the Denisovans. Further analysis of nuclear DNA showed that Denisovans were a sister population to Neanderthals. The two lineages shared a common ancestral population after separating from the ancestors of modern humans. They later diverged from each other, but they did not remain isolated. The Denisovan discovery transformed paleoanthropology because the physical remains were so limited. With most extinct human groups, scientists began with bones and later obtained genetic information. With Denisovans, the order was reversed. The genome appeared before the body. A few teeth and bone fragments provided additional clues. Later discoveries elsewhere in Asia expanded the picture. But no complete Denisovan skeleton has yet offered the kind of anatomical view provided by Lucy or Turkana Boy. Genetics had to carry most of the story. That story reached into the present. When researchers compared the Denisovan genome with the DNA of living people, they found evidence of interbreeding. Some populations in Oceania, particularly Papuan and other Melanesian groups, carry a significant amount of Denisovan-related ancestry. Smaller genetic contributions appear in several Asian populations. The pattern differs from region to region, suggesting that modern humans may have encountered more than one Denisovan population. One inherited Denisovan-related genetic variant is connected with adaptation to high altitude among Tibetans. A version of a gene involved in oxygen regulation appears to have entered the ancestors of Tibetan populations through ancient interbreeding. An encounter tens of thousands of years ago may therefore have contributed to survival in one of the most demanding environments on Earth. Ancient interbreeding was not simply a historical curiosity. It left biological consequences that still matter. Denisovan DNA also showed that extinction can be incomplete. A population may disappear as a recognizable group, while portions of its genome continue in mixed descendants. No living person is a Denisovan, but Denisovan genetic variants remain inside living human populations. The boundary between replacement and survival becomes difficult to define. Then researchers examined another bone fragment from Denisova Cave. The specimen was called Denisova 11. It was found among thousands of fragments that could not be identified by shape. Scientists first used protein analysis to determine that the bone came from a hominin. The fragment was more than 50,000 years old. Genetic analysis followed. The mitochondrial DNA was Neanderthal. But the nuclear genome revealed something extraordinary. Approximately half of the individual's DNA came from a Neanderthal population. The other half came from Denisovans. This was not a distant mixture inherited through many generations. Denisova 11 was the first-generation daughter of a Neanderthal mother and a Denisovan father. She was at least 13 years old when she died. The fragment does not tell us how her parents met. It cannot reveal whether they belonged to neighboring communities, whether one had migrated, or how mixed children were treated. But it provides direct evidence that two distinct archaic human populations reproduced together. Even her parents carried deeper histories. The Denisovan father had traces of older Neanderthal ancestry in his genome. The Neanderthal mother was more closely related to Neanderthals found elsewhere in the Altai region than to an older Neanderthal from Denisova Cave. Populations had moved across the landscape. They had replaced one another, returned, and mixed. The cave was not occupied by one stable group. It stood within a changing human world. Denisova 11 turned gene flow from a statistical pattern into an individual life. She was not a line connecting two branches on a diagram. She was a person whose parents came from populations that scientists once treated as separate categories. Her existence showed that those categories were biologically permeable. The same is true of modern humans and Neanderthals. Most people with ancestry outside sub-Saharan Africa carry a small amount of Neanderthal-derived DNA, although the proportion varies. Modern humans and Neanderthals interbred after Homo sapiens expanded into Eurasia. There may also have been earlier movement of genes in the opposite direction, from populations related to modern humans into Neanderthals. Human evolution was not a sequence of isolated species waiting to be replaced. It was a shifting network. Populations divided. They adapted to different environments. They became anatomically and genetically distinct. Then migration brought some of them together again. Contact could lead to competition, coexistence, cultural exchange, or reproduction. The fossil record rarely preserves those encounters. DNA can. That is why Denisova Cave belongs at number 1. The discovery did more than add another human population to a list. It changed the shape of the entire story. Laetoli showed that walking came before large brains. Lucy showed that old and new anatomical features could coexist in one body. Lomacui suggested that stone technology may predate the genus Homo. Turkana Boy revealed a nearly modern body built for movement long before the appearance of a modern brain. Dmanisi showed that small-brained humans with simple tools could leave Africa. Cima de los Huesos exposed deep Eurasian population structure and conflicting genetic histories. Homo naledi demonstrated that small-brained human forms survived surprisingly late in Africa. Homo floresiensis revealed an isolated Asian lineage unlike any human alive today. Jebel Irhoud pushed the beginnings of Homo sapiens deeper into the past and across a wider African landscape. Denisova Cave connected all of those lessons. Human evolution was ancient. It was diverse. It was geographically widespread. And its branches repeatedly crossed. The familiar tree of human evolution is still useful. It shows ancestry and separation. But it can also mislead. Real populations were not permanent lines. They expanded and contracted. They divided and reunited. Some contributed DNA to others before disappearing. A better image may be a braided river. Channels separate, move across different ground, and sometimes merge again. Some dry out. Others continue. The water downstream carries traces from many sources. Our species is the only human species alive today that can create the illusion that the past was always moving toward us. It was not. For most of human history, Earth contained more than one kind of human. Some were close relatives. Others belonged to lineages separated for hundreds of thousands of years. They walked, made tools, raised young, adapted to unfamiliar environments, and sometimes had children together. We are not the inevitable conclusion of their world. We are what remains after most of its diversity disappeared. And the evidence is still incomplete. Many regions of Africa and Asia remain poorly sampled. Tropical climates often destroy ancient DNA. Coastlines used by migrating populations now lie beneath the sea. Fossils sit unidentified in museum collections. Fragments too small to recognize may still contain proteins or genetic material from human groups unknown to science. The next discovery may not be a complete skeleton. It may be a tooth, a layer of sediment, a broken tool, or another piece of bone small enough to rest on a fingertip. What matters is not its size. What matters is the question it allows us to ask. Because every time scientists develop a new way to read the past, human history becomes less like a simple march toward the present, and more like what it truly was. A world of many humans connected in ways we are only beginning to understand.